In December 2023, the FDA approved the first therapy based on CRISPR gene editing, Casgevy, for sickle cell disease and beta thalassemia. These are painful, life-shortening inherited blood disorders affecting millions of people worldwide, predominantly in sub-Saharan Africa, the Mediterranean, and South Asia. For the first time in history, a tool that directly edits the letters of the human genome was approved as a medicine. This is not science fiction, this is now. Let's understand what CRISPR does in the clinic and where this technology is heading. We covered CRISPR-Cas9 in detail in Series 1, Video 7. To recap, CRISPR uses a guide RNA molecule to direct the Cas9 protein, a molecular scissors, to a precise location in the genome, where it makes a targeted cut. The cell then repairs that cut, and depending on the repair pathway used, the result can be a gene disruption or a precise sequence change. What makes CRISPR clinically revolutionary compared to earlier gene editing tools like zinc finger nucleases is its simplicity. To retarget CRISPR to a new gene, you simply design a new guide RNA, a short RNA sequence that takes about a day to synthesize. Earlier tools required weeks of complex protein engineering per target. Sickle cell disease is caused by a single mutation in the HBB gene. The mutant hemoglobin polymerizes under low oxygen conditions, distorting red blood cells into the characteristic sickle shape, causing them to block blood vessels, triggering excruciatingly painful vaso-occlusive crises and organ damage. The Casgevy approach is ingenious. It doesn't correct the HBB mutation. Instead, it exploits a biological workaround involving fetal hemoglobin. Every human produces fetal hemoglobin before birth, which does not have the polymerization problem. After birth, a gene called BCL11A acts as a switch that suppresses it. The CRISPR edit targets a specific enhancer sequence within the BCL11A gene, silencing it, which reactivates fetal hemoglobin production. The process. The patient's blood stem cells are collected from their bone marrow. The cells are edited with CRISPR ex vivo in the laboratory. The patient receives chemotherapy to destroy remaining bone marrow cells. The edited stem cells are then infused back into the patient to engraft and reconstitute the blood system. In clinical trials, 29 of 29 patients with sickle cell disease who received Casgevy had no vaso-occlusive crises for at least 12 months following treatment. For patients who had been hospitalized repeatedly for years, this represented a transformative change in their lives. Casgevy is an ex vivo therapy, but for many conditions like liver or neurological diseases, we need to deliver CRISPR directly into the body, in vivo editing. The challenge is delivery. How do you get the guide RNA and Cas9 protein to a specific tissue inside the body? Several approaches are being developed. First, lipid nanoparticles, or LNPs. They naturally accumulate in the liver. CRISPR components packaged in LNPs have achieved extraordinary results for liver diseases. In trials for transthyretin amyloidosis, a single IV infusion of CRISPR LNP reduced the disease-causing protein by over 90%. Then there are AAV vectors, which can carry CRISPR components to tissues like the eye, muscle, and CNS that LNPs cannot efficiently reach. Newer variants like base editing change a single DNA letter without cutting. Prime editing acts as a find and replace for the genome, capable of complex edits without double-strand breaks. The central safety concern with CRISPR is off-target editing. The guide RNA occasionally directing Cas9 to cut at unintended locations with similar sequences. If an off-target cut occurs near an oncogene or a tumor suppressor gene, it could theoretically contribute to cancer development. Modern tools to minimize this include whole genome sequencing of edited cells, high-fidelity Cas9 variants, and careful guide RNA selection using computational predictions. So far, clinical trial data has been reassuring. No cancer signals have emerged in thousands of treated patients, though long-term follow-up continues. Finally, the ethics. We must distinguish between somatic editing, treating a patient's own cells, and germline editing, editing embryos, sperm, or eggs. Somatic editing is broadly accepted. Germline editing is a different matter entirely, as changes are inherited by future generations. In 2018, a scientist shocked the world by claiming to create CRISPR-edited babies. The global community condemned the experiment as premature and unethical. The consensus is that germline editing should not proceed until safety is proven and society consents. The potential to cure disease is real, but we must navigate the ethics together. For more stories on the intersection of medicine, ethics, and policy, visit unbedsidemanners.com.